Good morning, everybody. Let me get rid of this. All right, so this first class period, we have actually just open for questions, exam recap, 3-point cross practice, any of that jazz. And also have a very loud complaining cat over there somewhere. So you may hear her yowling for a bit. So, who wants to go first? Let's see who has anybody has any questions. Otherwise, I have a 3-point cross practice that you can do. Because there will be at least one question on that, which is going to be a multi-question question. And in the meantime, Professor Luna is making sure I'm doing a good job. Aren't you? Not entirely sure what's going through her head. Any questions about the material that's going to be on the exam? So anything that doesn't make sense that you want to go over again? I'm scared for my life. It's not going to be that bad, I hope. Okay. Well, you're in luck, Rose, because I have one that I worked out this morning. So, shall we do that? All right. Well, before we start, what is Professor Crook's patented or patent pending 5-point plan for 3-point cross success? What's the first thing you look for? Find the double crossovers. Then what? What next? That's a good start. What do the double crossovers tell you? Gene order. Exactly. Then what do you do? Yeah, so that's what the double crossovers tell you, is which is the middle allele, because that's the one that has switched. Okay, so once you've figured out the double crossovers and the gene order, what do you do then? Single crossovers, right? One at a time. And then the final thing to do is draw a map, right? Okay, so oh, there you go. Thank you, Dipper. Exactly. Cannot forget the double crossovers in calculating the recombination frequency. Yeah, I think it actually ended up being like a 4 or 5-point plan. I kind of lost count after a while, but you get the idea. Okay, so make sure I don't set my phone. Oh, fun finding. Walking the dog, found a purple expo marker. So now we have 4 colors to work with. I was pretty excited about that. So, doopy-doopy-doo. 3-point cross practice. Oh yeah, draw a pretty map. So let's put up the categories. I got so much dog hair on this thing. And so move this back a touch. All right, so numbers are as follows. So here we don't have a simple all cis linkage phase going on. It's a little more complicated. So it should push you a little harder. So I'll let you copy that down. I think you can see all of it. And then we'll start working it out. We'll have a shot at doing it on your own first, Rose, and then we'll go through it all together. And so you can either confirm your results then, or you can get help getting your results. It's kind of up to you. So, Adam, what are double crossovers? Are they the most prevalent or the least prevalent, or somewhere in between? Nope. The opposite. The least prevalent, right? Because they're a result of two independent events both occurring. So they will be the rarest. Okay, what are the most common? Actually, question to the class. What category would these, what would these be? Yeah, those are the parentals. Thank you, Izzy. So this shows you what the original chromosomes look like. These all must be the result of recombination. And I don't know the answers because I just wrote this down this morning before lecture, so it'll all be new to me too. Okay, I'll give you another like 4 minutes or so. Okay, does anybody want more time or should we go through it? All right, I'll take that as well. Let's go through it. Okay. So here are going to be a few tips and tricks rows that will help you. So one way you can do it is once you worked out the gene order, you can change all of these to match that. Makes it a little easier. Or once you get the gene order, you can work out what crossover results in what combination of alleles. That's the other way of doing it. That's the way I typically do it because I'm a bit lazy. Don't want to rewrite all this stuff. So first up is gene order. Well, first up is figure out what are the double recombinants. So which ones are the double recombinants first? Which one's on here? 4 and 6, right? So these are the double ones. Okay, so now we compare these to these. right? So our original parental for one of them is plus C, right? And so if we look at this one here, we have A plus C, right? So A is moved. Double check with this one. This is AB plus. And so if A moves, we should have plus B plus, right? So look down here, that's what we have, right? So A must be in the middle. And as always, you can never assume that the gene order you're given is the correct one. Okay, so that's step one. Step two. So the gene order has to be BAC or CAB. It doesn't actually matter which way around. Okay. So if we put these into the correct order, that will be, that's the gene order. So we're going to have plus C and BA plus. So the way that I got that, let's kind of leave my rubber off go. The way you got that is because in the double recombinants, the allele that is moving is the one in the middle. And so you look to find on these which allele has changed compared to the parentals. So essentially, actually, I just got to get this off completely. We can look at it as over there, right? That plus C is the parental, and A plus C is the double. Right, so the one that's moved is this one. So if you write it out kind of in full, right, it would be plus C parent, and then the other one would be BA plus parent, and double crossover would be that. So it would be plus AC, and the reciprocal would be different color. That, which would be B plus plus. And that's what those are. How about that? Did that make a bit more sense? Let me just take a picture. It doesn't matter what order these are in the data that you get. Well, it matters what combination of alleles you have in the parents, because that's how you're going to figure out which one's moving, right? And then you need to know which ones are the rarest, because those are the double recombinants. And so you take what alleles are present in the parents, which are these ones, plus plus C or AB plus. And then you go, I think A must be in the middle because it's the only one moving. Right? For example, if these were the lowest number, you would say that C was the one in the middle. Give me a second, Adam. Right, because C is the one that's moving. That will give you plus, plus, plus and A, B, C. But it's not, it's these ones. And so if you want to double check, right, so this shows you that the A allele is the one that's moving in the double recombinant, so it must be in the middle. Rewrite them in the correct order. This hopefully will answer your question, Adam. That will give you plus plus C and then BA plus. And then the colors basically show you the result of the double recombination. Got a recombination between plus and A and between A and between B and A and between A and C, and that will get you these combinations, which are the double recombinants. So it does matter what order that you put the alleles in Galen once you work out the gene order, because that's the correct order of the genes. And sometimes as well, it actually helps writing these out in the correct order. And this kind of gets back to what Rose was asking about at the beginning, because then you can figure out what combinations of alleles do you get with a single crossover, right? We'll get to that in a second. Does anyone have any more questions about the whole gene order thing? Pardon me. Okay, so I'm going to rub this off and then put it further back up. So gene order is going to be B, A, C. I'm going to keep it in that order because it makes less changes essentially. And so if we rewrite the parentals, we're going to have, what would it be? Plus plus C, parentals B and P, and BA plus. Now, if you want to find out the distance, which is the third step, let's say between B and A, here, you're going to look for single recombinants will be B plus C, and literally just follow the line, right? Go from down here, up to here, and across. That's a result of a single crossover, so it would be B plus C, and then plus A plus. And now you go and look for which ones these match, right? And again, the order is going to be different, but it doesn't matter now because you know which letters you're looking for, right? Let's just get that off. And that's these ones down here. So the recombination frequency equals what? What do we add together and what do we divide it by? So Maria, that's correct. Single recombinants, which are going to be 31 and 28. Double recombinants, which is going to be 10. So it's going to be 31 plus 28 plus 10 over. Has anybody added all this up together? Because I haven't. Didn't get that far. 830. Times 100. That's down there in the shadow of my couch cushion. Everybody get where we got those numbers from? Okay, so what's the answer? Ten was from the double recombinants. Cool. And you always have to add the double recombinants because they still contain a crossover between those two genes. Yeah, I don't know, Galen, is that right? Oh, sorry, not Galen, not Galen, Galen. Maybe. Awesome. Okay, so 8.31 centimorgans, or 8.3 is enough for the peeps we ride with. Cool. Okay, so make a note of that. And now I should have left that one up there. Now if we have a crossover between, oh, Izzy, what did you get? So it's going to be 5969 over 830. Yeah. Blame my bad handwriting. I think you'd be on pretty steady ground if you did that. Okay, so what allele combinations are we going to get as a result of a crossover between A and C? What you gonna get? What you gonna get when they come for you? All right, what are our single crossovers going to look like? It's kind of easy because it's the ones that we haven't used yet, but still worth working out from first principles.
I'm still kind of confused on how to get those.
Which ones?
The crossovers, like I was kind of confused on how to get the A++. So what are we multiplying again?
So the A++, that is the result of, so basically it's actually easier when I'm doing it in class, obviously, because I can just watch my finger. The crossover between these two, right, will generate that chromosome. And it will generate the reciprocal, which is going to be that chromosome.
Oh, okay. Let me write that.
Mm-hmm. And there's absolutely no problem just literally using your finger, following it along. That's what I always do in class.
Oh, okay. I see that.
Yeah.
Okay.
Cool. It seems a shame to kind of rub that out, actually. It's kind of pretty. Now, when we do the same, all right, Marissa, see if you can figure that. Give me a second, Rose. We'll get to you a second. Okay, what allele combinations will a crossover between A and C generate?
Will it be the?
Did you get that?
Yeah, I do see that now.
And then the reciprocal would be that. Yeah, so we would have plus 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 BAC. And then we look over here, that corresponds to these. So we're looking at that. And then we can answer Rose's question. How do you determine which two genes? That's the distance. And so you're just looking for change in allele combinations, Rose. Right. So anytime you get this allele combination or this allele combination, it has to be the result of a crossover between A and C. And so the distance between A and C equals frequency of getting this single crossover plus the frequency of getting the double crossovers. That's pretty much it. So in this case, it would be 110 plus 113 plus our doubles over 830, which should be what? Did that make sense, Rose? Good. So this, what I'm doing here, this is a good, and this is exactly what I do too, right? So for the question in the exam, I actually worked it out myself, and this is how I worked it out, right? So this isn't any different from what I do. I'm not teaching you like dumbing it down or anything. This is what works, right? These orders aren't correct, so it's actually quite confusing trying to figure out what genes are being, what alleles are being swapped just looking at this stuff, right? That's why it's a really good idea to write out the gene order and then like the two different parental chromosomes, these ones, in the right order. And that will help you figure out, okay, if I have a recombination between these two, I'm going to get a certain combination of alleles. What do those look like on here? Ah, they're those ones. If I go recombination between these two, what alleles will that generate, or combinations of, right? It will generate these ones, look back here, see which ones match that, right? That's the most straightforward way. As I said before, obviously you can just rewrite all of this out in the correct order. That would also work, right? So we could just change this around and write BAC instead, for example, because that's the correct gene order. That might make it a little bit easier to figure out where the crossing over is occurring, but you don't have to, right? Those are the two different ways of figuring out what is getting crossed between what. Okay, what's the answer? .1. Yeah, round up to single decimal point. Although if it's to do decimal points, that's fine, but you'll see what the answers are in the exam. So you're just going to go with that. Okay, so let me take a picture of this first. That's there. So now all you do, make that a little bit bigger. And so our map is now B. Actually, crap, is it B and A was 8.3, right? To scroll up. Good. And 28.1. And there you go. That's all there is to it. So any questions, anyone left behind or not quite following what we did? And the plus side is if you can do a 3-point cross, then 2-point cross is a piece of cake. So you won't need to draw a map for the exam, but you will need to either select the correct gene order. Well, you'll need to essentially any question you're going to get on a 3-point cross is going to ask you, what's the gene order? You're going to have to select the correct one. And what is the map distance between at least 2 of the 3 genes? And then perhaps a nice easy one is whether or not linkage is occurring. So obviously the correct 2-cross one for sure. Obviously the correct answer is going to be in there. You just have to figure out which one it's going to be. Okay, 2-point cross. So we're going to deal with 2 genes. One is going to be solid color and round. Solid color, round. The other one is going to be square and empty. So this is going to be a heterozygote, which is going to be round, round, black or black. Right, so that's test cross setup. How do we find out they're linked again? If the recombination frequency is less than 50%, or the map distance is less than 50 centimorgans. But don't forget, also, if it's close to 50%, right? So anything that is because obviously you're going to have noise, right? So, you know, if you essentially what you're looking for is roughly equal number of parentals and recombinants. If you have an excess of parentals or deficit of recombinants, which is essentially the same thing, then linkage is occurring. So in the previous example, both those pairs of genes were linked. And actually all 3 of them were linked, to be honest, because the total recombination, the total map distance was less than 45 centimorgans. Does that make sense? So in this one, we're going to have these 4 different combinations. Parentals, recombinants. What's the map distance between R and B? R and B? Next time I should do C&W so I can talk about country and western. Those are parentals because two reasons. One, they're what you see in the parents. And second, more importantly, they are what are more prevalent. So if I were to be kind of even more particular, this would be our, well, let's do it in the proper way. Plus plus over RB, and this would be RB over RB. So this being in the cis linkage phase. If this was trans linkage, then the parentals would be very different. Does that make sense, Adam? Yeah. The shortcut way of figuring out which ones the parentals are in any genes that are linked are parentals are simply the ones that are most common. But you can also go and look back at the genotype and the chromosomes in the parent that's heterozygous. That will also tell you which ones are parentals. And in a back cross, you're always going to be crossing a heterozygote with homozygous recessive. So what's the map distance? Okay, how did you get that, Maria? What's your math? So we have 16.8 centimorgans, commons divided by the total, so that's, oops. And total is 415, 499, I think. So that's 84 divided by 499. Yep, times 100 equals 16.8. There you go, that's the distance between R and B. Now just be aware though that the exam 2 is not going to be just on linkage, okay? It's going to be on everything that we've covered since the end of exam 1. That's right, Dipper. It will be, again, like the other one, it will be in one question at a time, and you can't go back. Yeah, I know that's not ideal, and kind of my apologies for that. It's the downside of doing exams online. Oh, you're welcome, Adam. Cool. All right, everybody. So, need up. Yeah, that always helps. Finishing up a little early. Shall we meet at 12:10? Cool. All right. Neat. I shall post these up after class. Don't forget the exam opens at 1:30 and it closes at midnight tomorrow night, Wednesday. Okay, I'll just type that up. Yes, Maria, there will be lab today. Hopefully it's not going to take too long, so it shouldn't be too much of a drag, but there will be lab today. We'll figure out start time when we get through the next lecture. Maybe we can finish a little early, which would be nice. What's the date tomorrow? 7th. Will what be a new one, Adam?
So like what I'm meaning is, are we still going to be going over any questions that we have pertaining to the lab report in today's lab?
Yeah, we can do, for sure.
Okay.
Because that is due tonight at midnight.
But we're not starting a new lab that's due Thursday.
We will.
Oh, okay.
So yeah, I know, and it just never stops. Unfortunately, it's a constant avalanche of stuff. Yeah, today we'll be looking at DNA isolation, so how we go about doing that, and the assignment for that will be due Thursday midnight. So yeah, fun never stops, unfortunately, or fortunately, depending on your point of view. Cool. All right, everybody, see you back in 15 minutes. Galen might be a little bit earlier, depends on how much time we spend in the next lecture. So we'll figure that out, you know, at the end of lecture 2, which will be in about 15 minutes time. Cool. All right, go stretch your legs, see you in 15 minutes.